Just a second. Please move from the background. Okay. Is the camera angle fine? Yeah, okay. Is it fine now?
You are not visible.
I'm not visible,
Yeah.
but I can see myself in the laptop. So it means my camera must be okay. Okay, now? Am I visible now?
Yes, you can.
Is it fine?
Yeah, it's fine. You
Okay,
can
so
start.
the only issue is that I'll not be able to write, okay?
Okay.
Just the same as the other day. So should I like begin with the class?
Go ahead.
Okay, so I'm starting with the class then. Okay. Good evening. Myself, Dr. Beenish, and I'll be taking your biochemistry lives. For today, I'll be talking on glycolysis. Before I start with that, I'd like to introduce you to the core features of glycolysis. Core features of Oracle. So in Oracle, you have live classes, recorded classes, and you also have handwritten notes and MCQs. You have, apart from the academic support, you'll also have non-academic support like counseling, which will be like one-to-one counseling, very personalized. We have different subscription plans. The basic one, which is for 3 months, the standard is for 6 months, and the premium plan is for 12 months. So depending upon your needs, you can avail any of the three subscription plans. And already we have discounts going on, but you can have
Personalized.
20% discount by using my promo code that is displayed on the screen, DRBEENISH 20. Okay, so now I'll be taking the class. Due to some technical error today, I'll not be able to use my pen. We'll hope to resolve that in the next classes, so please kindly bear with it. Okay, so let's begin with glycolysis. Glycolysis is basically derived from two different words, glycas and lysis. Glycas means sweet sugar and lysis means cutting. So you can simply define glycolysis as cutting up of glucose or sweet sugar. Now, when do you think glycolysis will take place? It takes place in the well-fed state. Right after you've had your meal, the body is rich with glucose. That is when glycolysis will take place. So the hormone for well-fed state is insulin. Insulin is released only when there's glucose influx. So glycolysis takes place in the well-fed state under the influence of the hormone insulin. The site for glycolysis is cytoplasm, and glycolysis takes place in all the cells and all the organs of the body. Before beginning with the detailed steps of glycolysis, I'll just give you a brief overview as to what is happening in glycolysis. In glycolysis, glucose, which is a 6 carbon compound, is broken down, split into two 3 carbon compounds. You can see it is split into two 3 carbon compounds, and eventually two same 3 carbon compounds are formed. Okay, like I've shown you here with the arrow, they're split into two 3 carbon compounds, and that two 3 carbon compounds are changed into the same similar one 3 carbon compound. So you have two 3 carbon compounds which are same now, which changes into pyruvate. Now, depending upon the presence and absence of oxygen, pyruvate can have two different fates. It can undergo aerobic glycolysis in the presence of oxygen, leading to the formation of acetyl-CoA, and it can also, under the absence of oxygen, form lactate, which is known as anaerobic glycolysis. Now I'll take these steps of glycolysis in detail. So glycolysis can be divided for our understanding into two phases, preparatory phase and the payoff phase. Preparatory phase is where it's preparing itself to generate ATPs. Okay, so it is consuming ATP in that phase, and the payoff phase is where your ATP is generated. Now, again, the preparatory phase can be split up into two stages, the phosphorylation state and the splitting up stage. Now, phosphorylation, as we all know, is the addition of phosphate to a compound. Okay, so when you add a phosphate group to anything, that process is known as phosphorylation. And in enzymology, you must have studied that phosphorylation is brought about by enzymes that are known as kinases. Okay. So in the first step, that is the phosphorylation stage, we will see that your glucose, which has entered into the cell. Now, from where this glucose is coming and how does this glucose enter into the cell is a different topic in itself. But just so you understand, I'll give you a quick flashback. You've had your meal. The meal is rich in all sorts of carbohydrates, complex carbohydrate. It just does not have glucose only. Okay, so your food is broken down into simpler monomeric form, and ultimately everything that you've had, like your oligosaccharides, polysaccharides, disaccharides, everything's just broken down into its monomeric form, glucose. Okay, so this is where your glucose is coming from. Now this glucose is absorbed into the bloodstream. From the bloodstream it enters into the cell. Now how it enters into the cell is a vast topic, but for your understanding, as of now we have different glucose transporters from GLUT1 to GLUT14 depending upon on what side and where in the body it's present. So, for example, GLUT4 is present in your skeletal muscles. It's GLUT1 is present in your neurons. Okay, so depending upon, there are 14 different isoforms of your glucose transporters. Now, in the blood, this glucose which was present, it enters into the cell through one of these glucose transporters. So this is where your glucose is coming. So glucose is present in the cell, and as I mentioned earlier, all of this glycolysis will be taking place in cytoplasm. So this glucose enters the cell and then it is phosphorylated. In simpler forms, you can say this glucose is chained with phosphate. Okay, so glucose via the enzyme hexokinase forms glucose 6 phosphate. So this is the chaining or trapping of glucose inside the cell so that it does not move out of the cell, because the transporters, like for example GLUT1, it is bidirectional. So the glucose that is entering the cell can very well move out of the cell. So just so the glucose does not move out of the cell, this is the significance of this step. This is known as trapping up of glucose. Now the glucose has been trapped inside the cell, and this is brought about by the enzyme hexokinase or glucokinase. Now what is the difference between the two enzymes? I'll just talk about it. And in this step, now you see our substrate, which was glucose, did not have a phosphate group. Now our product is glucose 6 phosphate. A phosphate group has been added at the position 6, at carbon 6 of glucose. So where is this phosphate coming from? This phosphate group has been taken from ATP. So one ATP has been, energy has been used here in this step. One ATP has been used and it has been converted into ADP. And the coenzyme for this enzyme is cofactor is magnesium. So there are certain enzymes which show their activity in the presence of metals, which is known as cofactor. So magnesium is the cofactor here for the enzyme hexokinase. Now this was the significance of the first step, that glucose has been trapped. Now I'll quickly take the differences between hexokinase and glucokinase. Hexokinase and glucokinase, basically, they are different isoforms of the same enzyme. They are isoenzymes. Glucokinase is hexokinase 4 or D. So hexokinase is a housekeeping enzyme. Now, what are housekeeping enzymes? They are basically those enzymes which are functional all the time, whether in fed state or fasting state. They're just active all the time. And it is hexokinase is not induced by insulin, whereas glucokinase is inducible. It is induced by insulin. Now, hexokinase is inhibited by glucose 6 phosphate, whereas glucokinase does not show any inhibition by glucose 6 phosphate. Hexokinase has high affinity, which is also very obvious since I mentioned that it is a housekeeping enzyme. So it needs to be active all the time. So it needs to have high affinity for glucose. Therefore, it is working all the time. whereas glucokinase has low affinity. It is activated only when there is a large amount of glucose in flux. So it shows postprandial regulation, whereas hexokinase does not. And glucokinase is present in liver and pancreas only. And obviously, if something has an enzyme has a high affinity for a substrate, it has low Km. What is Km is your homework if you do not know. Okay. So this was briefly about the two enzymes. Now moving on to the second step. Now you see glucose 6-phosphate has been formed. Now this glucose 6-phosphate changes into fructose 6-phosphate. This is simply an isomerization reaction. Nothing much is taking place here. Only the glucose, which was the aldehydic form, has changed into the ketonic form. Okay, so the glucose changes into fructose, and this is brought about by the enzyme phosphohexose isomerase. And one more thing, in the previous step, when glucose 6-phosphate was formed, uh, one thing important that you should note is that this glucose 6-phosphate here has been trapped for metabolic purposes and not just simply glycolysis, because this glucose 6-phosphate even has other fates. It can enter glycogen synthesis, it can enter HMP shunt, it can enter into uronic acid pathway. And one of the pathways is glycolysis. Okay, so the glucose which has been trapped in the first step was trapped for metabolic purposes. And this first step was irreversible. An irreversible step of any pathway is called the flux generating step. Okay, now we see fructose 6-phosphate has been formed. Now the next step is that this fructose 6-phosphate changes into fructose 1,6-bisphosphate. Now this is brought about by the enzyme phosphofructokinase 1, and you need to always mention 1 because there's another enzyme phosphofructose kinase 2, which is very different from this one. So you need to mention 1. Now, this is one of the regulatory steps of glycolysis, and this is one of the regulatory enzymes of glycolysis. Very important. This forms the bottleneck of the reaction, and this is the committed step because fructose 1,6-bisphosphate that is formed does not have any other fate but to enter glycolysis. And here again, the cofactor is magnesium, and one ATP molecule has been used because in this step, this fructose 6-phosphate changes into fructose 1,6-bisphosphate. So one phosphate group has been added to position 1 of fructose. Okay. Now comes the moving on to the next step. This fructose 1,6-bisphosphate, which is a 6-carbon compound, it's split into 2 3 carbon compounds. One is dihydroxyacetone phosphate and the other one is glyceraldehyde 3-phosphate. Now this is brought about by the enzyme aldolase. Aldolase has two forms, aldolase A and aldolase B. So I've simply written aldolase because both of them are involved in glycolysis. Now, this dihydroxyacetone phosphate isomerizes into glyceraldehyde 3-phosphate. It happens so because the next enzyme in the cycle can only act on glyceraldehyde 3-phosphate. So this isomerization reaction is brought about by phosphotriose isomerase. Okay, so this DAP changes into glyceraldehyde 3-phosphate via the enzyme phosphotriose isomerase, and this is a reversible reaction. And also this splitting up reaction is reversible. The two DAP and glyceraldehyde 3-phosphate, they can join up again and form fructose 1,6-bisphosphate. Okay. Now aldolase is a lyase. Okay, you must have studied in the enzyme chapter that lyases are those enzymes which break covalent bonds without the use of water. Okay, so here aldolase is a lyase and it does not belong to the class hydrolases. Okay. Now moving on to the next step, glyceraldehyde. Now from here on in the cycle, you will have everything will be doubled twice for the simple reason because we have two glyceraldehyde 3-phosphate molecules because the DAP has also isomerized into glyceraldehyde 3-phosphate. So everything you will have to double it up in the cycle from now on. Now this glyceraldehyde 3-phosphate changes into 1,3-bisphosphoglycerate. So you see there has been addition of one phosphate group. Now where is this phosphate coming from? This is the inorganic phosphate which comes from the cytoplasm. And the enzyme used here in this step is glyceraldehyde 3-phosphate dehydrogenase. Okay, so this is the first oxidation reduction reaction taking place in the cycle. And dehydrogenases are those enzymes which remove hydrogen from one substrate and add on to the other substrate. So the hydrogen which this enzyme removed from glyceraldehyde 3-phosphate, it needs to be received, accepted by a reducing equivalent, which here in this case is accepted by NAD+. So NAD+ accepts the hydrogen which is removed by the hydrogenase enzyme to form NADH. Now this NADH enters into electron transport chain to form ATP. Now, one NADH will give you 2.5 ATP. So, as I said, that everything needs to be doubled up because you have two glyceraldehyde 3-phosphate molecules. So, there will be two NADH produced. So, you will have a total of 5 ATP molecules, okay? And also another importance of this step is, apart from ATP generation, that there's also generation of NAD+, which is very important for the continuation of glycolysis. Okay. Now moving on to the next step. This 1,3-bisphosphoglycerate changes into 3-phosphoglycerate, and this is brought about by the enzyme 1,3-BPG kinase. So this is the only reversible kinase. And here you see in the substrate, there were 2 phosphate groups. But in the product which is formed, there's only 1 phosphate group. So 1 phosphate group has formed ATP. That phosphate is received by ATP molecule to form an ATP molecule. So this is substrate level of phosphorylation, because we can see here at the level of the substrate, only one ATP molecule has been formed without the requirement of the electron transport chain. So we say that 1,3-bisphosphoglycerate is a high energy compound because you get one ATP molecule from it. Now, I'll talk about the arsenic poisoning point during inhibition. 1,3-BPG kinase can be inhibited by arsenate, okay? Because arsenic poisoning, in arsenic poisoning, what happens is your arsenic, it competes with the inorganic phosphate for its binding to the dehydrogenase. So this step here, where this ATP was generated, this step is bypassed, okay? So the glycolysis cycle will continue, but the ATP generation is not there in arsenic poisoning. I'll deal with that in detail later on when we study inhibition. That was just here to let you know that this step can be poisoned by arsenic. Now moving on, 3-phosphoglycerate changes into 2-phosphoglycerate. This is simply an isomerization reaction, because you can see the phosphate which was present at the third position in 3-phosphoglycerate only shifts the second position. So this is brought about by phosphoglycerate mutase. Although there is transfer of phosphate group, but since it is within the same molecule, a mutase enzyme has been used and not a kinase, because kinases, they transfer phosphate groups from one substrate to another substrate. But since this is within the molecule, so a mutase enzyme has been used, and it is an isomerization reaction. Now moving on to the next step, 2-phosphoglycerate, which is a 3-carbon compound, it changes into phosphoenol pyruvate. This is again a 3-carbon compound. But what happens is 2-phosphoglycerate does not have any double bond. But here there's double bond, enol has a double bond. So it changes into a 3-carbon compound which has a double bond. So how is a double bond formed? By removal of water. So this is a dehydration reaction. So a water molecule has been removed from the single bond to form a double bond. And enolase also requires the cofactor magnesium. Now this phosphoenol pyruvate, again, enolase is inhibited by fluoride. I'll talk about it later. Now this phosphoenol pyruvate, it changes into pyruvate, but this is brought about by the enzyme pyruvate kinase. And again, you can see one ATP has been formed. So this is substrate level phosphorylation again, because ATP has been formed at the level of the substrate. Now this pyruvate can have two fates. It can either form acetyl-CoA, which enters into TCA, or it can form lactate. Now, when it forms lactate, this takes place when there is no oxygen, in the absence of oxygen. So this is anaerobic glycolysis. This step is very important. Anaerobic glycolysis is important in your RBCs because they do not have mitochondria, so there is no electron transport chain running there. So for the regeneration of NAD plus, which we had seen, it was required here in this step. So for the continuation of glycolysis, it is very important that NAD plus is regenerated. So in anaerobic glycolysis, this step becomes very important. And also, when does anaerobic glycolysis take place? It also takes place when skeletal muscles are exercising. Then also there's absence or low supply of oxygen. So to relieve the muscles of fatigue, uh, this takes place. And otherwise, anaerobic glycolysis usually does not take place because the ATP formed, we'll study in energetics, the ATP formed is low, therefore otherwise doesn't take place. Only when required, that is when your anaerobic glycolysis will run. Otherwise, in normal circumstances, it is the aerobic glycolysis, formation of acetyl-CoA, which enters TC, that is what usually takes place. So this was the steps of glycolysis. Now moving on. I've already spoken about this, but I'll take this once again. Significance of anaerobic glycolysis, as I mentioned in mitochondria, in RBC, where there's no mitochondria and no electron transport chain running. So that step becomes important because formation of lactate from pyruvate, it is coupled with glyceraldehyde 3-phosphate dehydrogenase enzyme. So regeneration of NAD plus is very important. Now moving on to the energetics. Now in aerobic glycolysis, we have already seen that NADH was formed, as I mentioned, 2 NADH were formed, so 5 ATP from there. And then from substrate level of phosphorylation, we get 4 ATP. There were 2 steps that were giving substrate level of phosphorylation, and we have to double everything up, so 4 ATPs. So a total of 9 ATPs formed, and we had consumed 2 ATPs in 2 steps for phosphorylation. So you have to subtract that. So a net total of 7 ATPs is formed in aerobic glycolysis. Now in anaerobic glycolysis, you do not get the ATPs from NADH. So the net ATP formed is only 2. Now moving on to the regulation of glycolysis. Now, as I mentioned earlier in the beginning of the class, that glycolysis operates in the well-fed state. So in the well-fed state, the hormone that is active is insulin. So, and insulin is the hormone which activates enzyme via dephosphorylation. So for an enzyme to be active under the influence of insulin, it has to be in the dephosphorylated state. So the regulatory enzymes of glycolysis, that is hexokinase, phosphofructokinase 1, and pyruvate kinase, they're all active when they are dephosphorylated. Now moving on to the allosteric regulation. Hexokinase is inhibited by glucose 6-phosphate. Phosphofructokinase 1, it is inhibited by ATP. That is when the energy is high, ATP is high. ATP is a signal of high energy in the cell. That is when your phosphofructokinase 1 will be inhibited. Also, citrate and low pH inhibits phosphofructokinase 1. An activator of phosphofructokinase 1 is AMP. AMP is a signal of low energy in the cell. Fructose 6-phosphate and fructose 2,6-bisphosphate. Fructose 2,6-bisphosphate is one of the most potent activators of phosphofructose kinase 1. Since my pen is not working, okay, I'll try to manage. So phosphofructose kinase 1 has another isoform, phosphofructose kinase 2. So phosphofructose kinase 2 is a bifunctional enzyme. It is a tandem enzyme which has 2 active polypeptides. Okay, it shows kinase activity and as well as phosphatase activity. So phosphofructose kinase 2, the kinase activity, when it is activated, it forms fructose 2,6-bisphosphate. Okay, like phospho, as we have seen earlier here. Yeah, this one. This phosphofructose kinase 1 was causing phosphorylation at position 1. Okay, so it forms fructose 1,6-bisphosphate. But phosphofructose kinase 2, it phosphorylates as position 2. So it forms fructose 2,6-bisphosphate. And this fructose 2,6-bisphosphate is a very potent activator of fructose 1, phosphofructose kinase 1. Now, pyruvate kinase, it is inhibited by ATP. Moving on to the clinical correlations, not many disorders are associated with glycolysis as it is the most important cycle. So, if at all any major disorders are there, the child will not be able to survive. Although we do have some minor deficiencies like pyruvate kinase deficiency, it causes hemolytic anemia. Hemolysis of RBCs will be there in that case. Phosphofructose kinase 1 deficiency, it causes muscle fatigue. The person will not be able to tolerate exercise. And our skeletal muscles, they have high glycolytic capacity. Therefore, skeletal muscles are able to tolerate ischemia to some extent, but cardiac muscles, they do not have high glycolytic capacity. So therefore, in case of ischemia, they do not tolerate it well. Now, another newly added clinical correlation is MODY, that is maturity onset diabetes in young, MODY2, and this is due to mutations of glucokinase, because glucokinase is working as a glucose sensor. Okay, since my pen is not working, I'm having trouble. Okay, here. Now, hexokinase was a housekeeping enzyme. It is active all the time. And I said glucokinase is inducible. So when there's a lot of glucose influx and it is present in your liver and pancreas. So when there's a lot of glucose influx, so to overcome that, glucokinase gets induced by insulin, because glucokinase has a higher Vmax, reaction velocity enzyme has a higher Vmax as compared to hexokinase. So hexokinase can only phosphorylate normal levels of glucose, but when in case of right after you've had a carbohydrate rich meal and there's a lot of glucose influx, to maintain the blood glucose level, that is when your glucokinase is active. So glucokinase helps in maintenance of blood sugar level. It is a glucose sensor. So when there is mutation in glucokinase, it leads to MODY. Do you understand how it's related? Okay, now inhibitors of glycolysis. Iodoacetate, it inhibits glyceraldehyde 3-phosphate dehydrogenase. Fluoride, it inhibits enolase. This finds one of the it has one of the clinical applications. That is when you are collecting blood for estimation of blood glucose, you add fluoride to the sample so that glycolysis stops. It is inhibited and you do not get lower values of blood glucose. And arsenate, it inhibits 1,3-BPG kinase, arsenic poisoning. How I just mentioned that it competes with the inorganic phosphate. Now you have one thing called Warburg effect. You might sometimes be asked about it. So I'll tell you briefly. In Warburg effect, there is a cancer cells, they utilize more glucose. There is oxygen present in the body, but still the body goes for uh, anaerobic glycolysis, forming lactate, although there is presence of oxygen. So this leads to more utilization of glucose since the ATP generated by anaerobic glycolysis is less. So to compensate that, it uses more glucose. So that is why it is said that cancer cells utilizes more glucose. And this is what is Warburg affected. And it also finds its application in PET scans. Okay. It is an imaging technique. You'll not be asked in so much detail. So basically, PET scan is an imaging technique where you differentiate between cancer cells and non-cancer cells. You're giving an radio-labeled isotopic form of glucose and then see which of the cells are using more glucose. So you get to know that these are cancer cells. Okay. So this was briefly about glycolysis, and some MCQs are there. I'm having some glitch, so I can't even go for polls. I think, yeah. Okay, I can't even have polls. So if you could see, true statement about glucokinase is R. The first option is Km value is higher than normal blood sugar found in liver. Glucose 6-phosphate inhibits it. has both glucose 6 phosphate and kinase activity, and glucose enter cells through GLUT2. Okay, so I've spoken about glucokinase. So the question says, which of the following statements are true? There can be one or more statements that can be true. Km value is higher than normal. Yes, that's true. Glucokinase is found in the liver. Yes, that's also true. Glucose 6 phosphate inhibits it. No, that was about hexokinase. Has both G6 phosphatase and kinase activity? No, that's about phosphofructose kinase 2. Glucose enters cell through GLUT2. That's also correct. So your option A, B, and E are correct here. So finally, with this, we come to an end of our class. It was little below than my expectations, since the pencil was not working and I was not able to explain things better. But hopefully we're looking to resolve this in our next live, and you'll be having your next session on the 17th, and I'll be talking about glycogen metabolism, both synthesis and lysis. And we hope to fix this glitch by then. It was nice having this session. Thank you.
हो गया
बहुत गंदा है,
हो गया?
अच्छा नहीं था।
हो गया खत्म?
आई डोंट नो।
ये क्या बन के आ रहा है? तो बहुत स्लो है वे रमन। उससे तो इतना स्लो वगैरह कैसे हो गया?
आई डोंट नो, जस्ट लीव